Perfect. All right. My name's Amy. I'm a third year PhD student in the Jackson Lab and the McDonald Lab at the University of Birmingham. I work very closely with my collaborators at CAVI and also the St Helena Research Institute. So today I'm going to be talking about tree disease on St Helena. So ooh, let me just, oh, there we go. My slide's moving. There we are. So as we all know, St. Helena is a small island situated in the South Atlantic, and its volcanic terrain means that many different ecosystems exist on the island. And along the central ridge, we have an endemic cloud forest that has become established. Within the endemic forest, there are several important species, some of which are listed here. So the she cabbage, dogwood, whitewood, black cabbage, and he cabbage are some of the iconic ones. And many of these are actually in the Asteraceae family. So that's the daisy family. However, in many threats exist to the cloud forest that we're already aware of, and these have been presented nicely already during this conference. So many of the species have few individuals remaining in the wild, but also in the field gene banks. And this is because of land clearance and grazing and the introduction of invasive species. So for most of these species, there's less than 100 individuals remaining. And the last IUCN Red List survey declared she cabbage, for example, now extinct in the wild. And as a result of the small population sizes, some of these trees are now sterile. They're producing very few or not viable seed. And this is resulting in inbreeding depression. Some of these species are also being forced to higher elevations, and this is a result of the land clearance. Along the central ridge, you typically only see the black cabbage trees as well as the endemic tree ferns. But because they're losing space, some of these trees might be now found outside of their natural range. And as a result of this, they might lack certain adaptations to live in these slightly different elevations. And they might also be impacted more so by other factors such as climate change. And as we've already mentioned, they might be might be exposed to competition because of these introduced non-native species. So the aim of my project is looking at these trees because the concerns over the future stability of them has risen due to an emerging unknown disease-causing agent. So not only mature trees, but also juveniles across the peaks, as well as the field gene banks and in the endemic nurseries, have been suffering from sudden dieback. So my project is looking at identifying a possible pathogen which could be a candidate for causing this disease, proving that we found the right candidate pathogen through pathogenicity testing, and then starting to try and characterize the pathogen population on the island. For this talk, I'll be sort of giving an overview of how we go about identifying a candidate pathogen, more so than going into the molecular side of stuff, but I'll introduce some of our recent molecular findings. And on this map here, I'm just showing some of the sites that I'll be talking about. So we have the George Benjamin Arboretum and Ginger Patch, which are well-established field gene banks. And then, of course, the Peaks National Park. So our first question is, how do we go about identifying a causal agent of disease? Well, we know that different pathogens can affect different areas of the tree. So in the phyllosphere, we have fungi, bacteria, viruses, mycetes. On the trunk of the tree, we typically see diseases caused by bacteria and mycetes. And in the roots, which are our top candidates are usually oomycetes and fungal pathogens. But this, of course, isn't, there can, of course, be diseases caused by many different microorganisms across the tree. So these are just some examples. So acute oak decline is a polymicrobial disease. This means that it's caused by more than one pathogenic species. And we have Phytophthora ramorum, which causes these similar lesions, but this is actually a oomycete pathogen. We also see Phytophthora ramorum causing needle blight, particularly on larch. And it's very similar to symptoms of Dophthaloma, which also causes an infection of the needles, but this is actually a fungal pathogen. And then when we look at the roots here, these are actually two examples from Australia. One is a pneumycete pathogen, Phytophthora cinnamomum, and the other is a fungal pathogen, which we can actually see working its way up the trunk of the tree. So when we put all this into perspective, when we're thinking about disease on St Helena, we can start to infer what might be going on. So initially, we see this sudden wilt and collapse of trees throughout the field gene banks as well as the Peaks National Park. We then go further down the tree and we see these necrotic dark areas around the collar, so the base of the tree. And once we uncover the root system, we can see a massive loss of the smaller roots as well as the lateral roots, and necrosis and staining of any roots that remain, which ultimately results in death of the tree. So to go about identifying a candidate pathogen from what we've seen, first we've made our observations. We can see we're probably working with a root pathogen. We then take regions of the root from the leading edge of the necrosis, and we put this onto selective media. So the media is providing nutrients to grow the microorganisms, and we can treat these with different antibiotics and antifungals to make it selective to different types of microorganisms. And what we noticed straight away was we were consistently isolating this morphotype. And we soon identified this as a pneumycete pathogen known as Phytophthora cowmanii. And what really worries us about a pneumycete pathogen is that unlike fungi, pneumycetes have an asexual reproductive stage where they produce zoospores. And these zoospores have flagella, which means the spores are able to move in water. And this is a big problem for the movement of pathogens through soil. So this can be through trade and transport, through contaminated soil on vehicles or even on your shoes, and also through the importation of contaminated goods such as crops, plants, and also trees. So now that we have a good candidate pathogen, we needed to prove that this was actually causing disease on the endemics. So we did this using a pathogenicity test on two different endemic species. So the whitewoods being one of the most severely impacted endemic tree species that's widely replanted, and dogwood, which unlike whitewood and some of the other endemic species, doesn't show that typical collapse and wilt that we saw in the field. And there are several mature individuals of dogwood left remaining in the wild. So to do this, we used a rice inoculation method. We incubate autoclaved rice with the pathogen. This means each rice grain has a nice covering of mycelial growth, and we then damage the roots of our saplings, and we put either 5 or 15 grains of rice into the soil with each sapling. We then left these and observed the below ground symptoms. So this is what we saw 5 weeks post inoculation. This is the whitewood roots. So we see a severe loss of the smaller roots. Our control saplings were treated with just plain rice. There was no pathogen introduced into the soil. Our low inoculum concentration was 5 grains of rice, and our high inoculum concentration was 15 grains of rice. And in this graph here, you can see that there's a significant difference in the root length of our high inoculum treated saplings compared to our controls. And interestingly, there was no development of disease in the dogwoods. Now, this really supports the idea that the sudden wilt and collapse symptom that we're seeing in the field is indicative of infection with this particular Phytophthora species and not a different pathogen. So now that we have our candidate and we've proven that it causes disease, we then wanted to look more closely at the pathogen population on the island. And to do this, we use some phylogenetic analysis. So we start off by whole genome sequencing. So this is just showing us unwinding our chromosome and revealing the nucleotides, which is the way DNA is encoded. We extract several different genes. So in this case, we used single copy orthologs. So these are highly conserved genes that should be present in all of our isolates. And then we carry out the phylogenetic analysis to build this sort of family tree. And this is just the schematic of what we found. So Phytophthora pseudocryptogaea is just a closely related species to Phytophthora kalmanii. And then we have our Phytophthora kalmanii. And then here we have our island isolates. So as you can see, they produce this step-like structure with each other. And actually, we could correlate this step-like structure to time. So this is actually the actual tree that we produced from this analysis. The numbers on the branches here are just the bootstrap value. So this is the statistical support for the relationship between each isolate that we sampled. And as you can see, we're nicely seeing the evolution of the pathogen, the diversification of the pathogen over time. And these isolates are from different sites as well. So this is all from the Peaks and the George Benjamin Arboretum, with 2 early isolates actually from the endemic nursery. So not only is this happening across time, but it's happening with it across multiple sites on the island.
About 5 minutes, Amy. 5 minutes.
Perfect, thank you. I'm nearly done anyway. So in conclusion, we can see that Phytophthora kalmanii is causing disease on at least one endemic tree species. And we know that the whitewoods are highly susceptible. In comparison, the dogwoods are not susceptible, but we know that they're still showing symptoms in the field. And this opens up a lot more questions than it answers. Is there more than one pathogen at play on the island, or are dogwoods maybe more susceptible to environmental changes rather than the pathogen? And we can start to see that the pathogen population is reflecting diversification over time. Some additional things that we've started to look at is more of the population genomics. So looking to see how the pathogen is mixing, whether sexual reproduction is at play, and now that we have a reference genome for the isolate, I've started to construct a pipeline to extract effectors from the genome so that we can start to look at the interactions between the pathogen and known resistant markers in different plants. So with that, I'd just like to thank everyone at Birmingham and at CABI and there on St Helena, also the Cloud Forest Project and the St Helena Research Bursary, which has enabled me to go to the island more times than I was meant to continue carrying out this work.